Okay, ladies and gentlemen, you are welcome. So in this video, we are going to look at MCQ on microbiology. Question 1. What is microbiology? A. Study of molecules that are visible to human eyes. B. Study of animals and their family. C. Study of organisms that are not visible to naked eyes. And D. Study of microscope. So the answer is C. Microbiology is a branch of biology that deals with the study of microorganisms, and these microorganisms or these organisms that we cannot see them with our naked eyes are called microorganisms. So therefore, as you can see from the explanation, the study of organisms that are not visible to naked eyes is known as microbiology. Even from the word micro means small, bio means life, logy means study. So the study of tiny life, that is organism that we cannot see with our naked eyes. So therefore, to see these organisms, a special device known as a microscope is required to view these organisms. And examples of these organisms, we have bacteria, viruses, and fungi. So these are the organisms that we cannot see them with our naked eyes except when we use microscope. So microbiology is a branch of biology that trying to describe that. Then 2, who is known as a father of microbiology? A. Edwin John Butler. B. Ferdinand Kohn. C. Robert Koch. D. Anthony van Leeuwenhoek. So the answer is option D. Anthony van Leeuwenhoek is a Delft cloth merchant and is considered as a father of microbiology. He's the first scientist that was able to discover invisible world of microorganisms using a homemade microscopes. He used a homemade microscopes to visualize the first microorganisms, and that is why he regarded as a father of microbiology. Question 3. Which organisms among the following perform photosynthesis by utilizing light? A. Cyanobacteria, fungi, and viruses. B. Viruses. C. Cyanobacteria. D. Fungi. The answer is cyanobacteria. Explanation. Cyanobacteria, they are organisms that require light as a source of energy to perform photosynthesis. And after all, they are green, they have chlorophyll, so they use sunlight as a source of their energy to undergo photosynthesis. But fungi and viruses are unable to perform photosynthesis, therefore they are called heterotrophic, while cyanobacteria are called autotrophics. Autotrophics meaning that they can synthesize their own food because they can undergo photosynthesis, while fungi and viruses, they are heterotrophic because they cannot synthesize or they cannot undergo photosynthesis and they depend on other organisms for their survival. So then question 4, which part of the compound microscope helps in gathering and focusing light rays on the specimen to be viewed? A. Condenser lens. B. Magnifying lens. C. Objective lens. And D. Eyepiece lens. So we have different types of light microscope, and examples of it we have compound microscope. And some of this microscope they have, or this microscope they have different components, and these different components have their own specific functions. So in a compound microscope, the part of the compound microscope that helps in gathering and focusing light on the specimen to be viewed is, the answer is option A, condenser lens. A compound microscope contains three separate lens systems. The condenser lens is placed between the light source and the specimen, and it gathers and focuses the light rays in the plane of the microscope with the specimens. So question 5, which of the following are produced by microorganisms? Alcoholic beverages, B. Fermented dairy product, C. Bread, and D. All of the mentioned, mean all of the above. So the answer is all of the above. Ladies and gentlemen, microorganisms, so many people are thinking that when we said microorganisms, they are thinking that they are dangerous, they only cause disease, but the truth is not like that. There are so many microorganisms that are of industrial importance. Like, for example, we can have a microorganisms that can produce alcohol. We have some that can produce dairy products. You can use some to produce bread. You can use some to produce cement. You can use some of the microorganisms to produce gold. So therefore, microorganisms are used in many industries for the productions of food we eat, like fermented dairy products, sour cream, yogurt. Like yogurt, we have Lactobacillus thermophilus are examples of the microorganisms that are found in yogurt, as well as a fermented food, as pickles, breads, and alcoholic beverages. So microorganisms, they are important in various industries. So question 6, what is the approximate size of the bacterial cell? Bacterial cell. So here we have different organisms, virals, human cells, and bacterial cells. So what is the approximate size of the bacterial cells? Please, let me see. Can you write this on the comment section before I give the answer? You have 3 seconds to do that. Tick tock, tick tock, tick tock. Yes. So the answer is B. The size of the bacteria are very small, most being approximately 0.5 to 1.0 micrometer in diameter. So this is the reason why they cannot be seen by naked eyes and can be observed under the microscope. So we cannot view them with our naked eyes because if you look at the size, it's very small, 0.5 to 1.0 micrometer means minus 6 meter. Okay, question 7. The greatest resolution in light microscopy can be obtained with A. Shortest wavelength of visible light used. B. Longest wavelength of visible light used. C. An objective with minimum numerical aperture. D. Shortest wavelength of visible light used and an objective with a maximum number aperture. So let's see. The greatest resolution that is used in light microscopy for we to see some of these microorganisms are what? The answer is D. So at this point, the explanation is that the relationship between numerical aperture, that is NA, and resolution is equal to wavelength divided by 2. Thus, maximum resolution is obtained with the shortest wavelength of visible light and an object with a maximum numerical aperture. So it means that you need a larger numerical aperture for the objective, and then you need the shortest wavelength. That is why the shortest wavelength of visible light used and an object and an objective with a maximum numerical aperture will be the best to give you the greatest resolution in light microscope. So 8. Which of the following is used in electron microscope? Electron beams and magnetic field, light waves, C. Magnetic field, D. Electron beams. Even from the word electron microscope, ladies and gentlemen. So electron microscope, it uses both electron beams and magnetic field to produce the image, whereas the light microscope uses light waves and glass lenses. So in electron microscope, a much higher resolution is obtained with extremely short wavelength of the electron beam. So we use electron beams and magnetic fields, magnetic fields to produce the image of a very tiny object, and that is why it needs a much higher resolution compared to the light microscopy. So that is why the wavelength should be extremely short. Question 9. Which among the following are spirochetes? A. Streptomyces species. B. Treponema pallidum. C. Spirillum bulatans. D. Cyanobacterium diphtheria. The answer is B. Spirochetes are flexible and can twist and contort their shape, whereas spirilla are relatively rigid. So Triphonema palladium belongs to a spirochete groups, and Spirillum volutans belong to the spirilla group. I think I can advise you here to go and watch my video on the different bacterial shape and sizes. Question 10. Bacteria having clusters of flagella at both poles of the cells are known as Amphithecus, Monothecus, and Perithecus. And then D, we have Lophothecus. So the answer is A. Explanation. In Amphithecus in Amphithecus, flagella occur either singly or in clusters at both cell poles. But for the Lophothecus, it refers to a cluster of polar flagella. And then for the Perithecus, it's surrounded by lateral flagella. And then Monothecus is for a single polar flagella. So therefore, in the case of the bacteria that having a cluster of flagella at both folds of a cells, is called Amphithecus. So that is why the A is the best answer. All these things is there in the general microbiology or general medical microbiology textbooks and material. So you can check the classification of bacteria based on their or based on the size of flagella. So 11, the respiratory chain of bacteria is associated with the A, cytoplasmic membrane, B, cell wall, C, cytoplasm, D, mitochondrial membrane. You know that bacteria, they doesn't have mitochondria, so therefore their respiratory chain is associated with the cytoplasmic membrane, and that of eukaryotic is present in the mitochondrial membrane. So therefore, only eukaryotic cells have mitochondrial membrane, while bacteria, they doesn't have because they are prokaryotic. Therefore, their respiratory chain is associated with cytoplasmic membrane. So glycolysis can occur -A, anaerobic cells, B, aerobic cells, C, neither aerobic and anaerobic cells, D, both aerobic and anaerobic cells. The answer is D. So glycolysis can occur either in the presence or absence of oxygen. So it is oxygen independent. So whether in aerobic and anaerobic, the presence of oxygen will not stop it, and the absence of oxygen will not stop it. So glycolysis does not require the presence of oxygen, therefore can occur in both aerobic and anaerobic cells. So in anaerobic cells, glucose is degraded into fermentation product, that is ethanol and carbon dioxide, and in aerobic cells, glucose is degraded into carbon dioxide and water. Question 13. Which of the following enzymes removes the RNA primer with its 5' nuclease activity? A, DNA polymerase 3, B, RNA polymerase, C, DNA polymerase 1, D, DNA polymerase 2. The answer is C. The DNA polymerase 1 is the enzyme that removes the RNA primer with its 5' nuclease activity. Simultaneously, it fills in the gap with DNA via its 3' polymerase activity. So this usually takes place during DNA replication in a cell. So question 14, phosphorus is the essential component of A, phospholipids, B, teichoic acid, C, nucleotide, D, all of the mentioned. The answer is D. Phosphorus usually supplied in the presence of phosphate. It's an essential component of nucleotides and nucleic acid, phospholipid, teichoic acid, and other components. So the phospholipids, the teichoic acid, and nucleotide, they are all major components. So they all require phosphorus as their major component. So question 15, the bacterium Staphylococcus, the bacterium Staphylococcus. Question 15, the bacterium Staphylococcus aureus is which type of bacteria? A, mesophile, B, mesophile and psychrophile, C, psychrophile, and D, thermophile. So these are the classification of bacteria based on the temperature requirement for them to survive. So the answer is E, mesophile. Mesophile are those bacteria that survive between 30 to 37 degrees centigrade, that is within a normal body temperature. You know that Staphylococcus aureus is a bacteria that are found in humans, so therefore they will definitely survive within the human body optimal temperature. So that's why it is called mesophile. So Staphylococcus aureus is a mesophilic bacteria which can grow in a temperature range of 6.5 to 46 degrees Celsius and has an optimal temperature at 30 to 37 degrees centigrade. So now question 16. The growth of bacteria or organisms are referred to as A, changes in a total population, B, an increase in number of cells, C, an increase in the size of an individual organism, D, an increase in the mass of an individual organism. The answer is B, an increase in a number of cells. So growth donates the increase in a number of cells beyond that present in the original nucleus, and it was not referred to as increase in size or mass of an individual organism. So growth is associated with an increase in a number of cells. That is the growth of bacteria or organism. So question 17, which of the following method can be used to determine the number of bacteria quantity? Which of the following method can be used to determine the number of bacteria quantitatively? A, spread plate, B, streak plate, C, pour plate and spread plate, D, pour plates. The answer is C. That is pour plate and spread plate. Due to the higher degree of dilution, pour plates and spread plate techniques might be performed in a quantitative manner to determine the number of bacteria present in a specimen. So these are the two methods, the pour plate method and spread method are the two methods that are used in quantitative determination of bacteria in a specimen. So question 18, which among the following are microaerophilic bacteria? Microaerophilic bacteria. A, we have Treponema. B, we have Borrelia. C, we have Spirocheter. D, we have Christispira or Christispira. B. And the answer is Borrelia. Borrelia is a genus of bacteria that belong to the class Spirochaetea and are microaerophilic. When we said microaerophilic, it means that they are bacteria that require small amount of oxygen to survive. That is microaerophilic bacteria. So this is actually classification of bacteria based on oxygen. So that is why, ladies and gentlemen, I want you to go and check some of my videos on this classification. Question 19. Penicillin causes inhibition of mycoplasmas. A, false. B, true. The answer is false because the mycoplasmas, the lack cell wall, and cell wall is cell wall is highly rich in peptidoglycan, and this penicillin is a compound or is antibiotics that use to inhibit the synthesis of peptidoglycan. So because of the lack of cell wall, mycoplasmas are not inhibited by even high levels of penicillin. However, they can be inhibited by antibiotics that affect protein synthesis, such as tetracycline or chloramphenicol. Chloramphenicol. So you see, because the penicillin is only targeting the cell wall, that is peptidoglycan cell wall. So that is why, because mycoplasmas, they doesn't have peptidoglycan cell wall, so that is why penicillin cannot inhibit their growth. So now question 20. What are the major cell wall structural component of fungi? A. Peptidoglycan, B. Cellulose, C. Chitin, D. Chitin, cellulose, or hemicellulose? The answer is D. So the cellulose structural component of fungi are chitin, cellulose, or hemicellulose, whereas the bacterial cell wall is composed of peptidoglycan. Question 21. Cryptococcosis, cryptococcosis is a disease of viral infection. B. Mycotic infection. C. Parasitic infection. D. Bacterial infection. The answer is B. It is a disease of fungi. So that is why it is called mycotic infection. And Cryptococcus neoformans is an important basidiomycetes pathogens of humans causing cryptococcosis, a generalized mycotic infections involving the bloodstream as well as lungs, central nervous system, and other organs. So Cryptococcus neoformans is an example of the fungi. Is an example of fungi that cause a disease in human called cryptococcosis, and it is affect the bloodstream, lungs, and the central nervous system and other organs in humans. So 22, which among the following is a pathogenic algae for humans? A, we have Cephalosaurus. B, we have Acanthopeltis. C, we have Chlorella. D, we have Prototheca. So the answer is D. Prototheca has been found to be a probable pathogen of humans, and it has been found in systematic and subcutaneous infections as well as in bursitis. Question 23. Chrysolamine is in the reserve food of A. Bacillariophycophyter, B. Xanthophycophyter, C. Chlorophycophyter, D. Heophycophyter. The answer is B. Chrysolamine is the reserve food of Xanthophycophyta, which is the yellow-green algae along with oils. Protozoa that eat other organisms are known as A. Parasitic, B. Mutualistic, C. Holozoic, D. Saprophytic. The answer is parasitic. The answer is Holozoic. So Holozoic protozoa are protozoa that eat other organisms. Species of Paramecium and are holozoic, and they must have a supply of bacteria or other protozoa. So this is called holozoic. Plasmodium divide by which of the following method most commonly? Plasmodium divide by which of the following method most commonly? A. Regeneration, B. Budding, C. Binary fission, and then D. Multiple fission. The answer is D. Multiple fission. The malarial parasite Plasmodium divide by the process of multiple fission, where it is known as schizogony and and serves to spread the parasite quickly in the host. Which of the following is a family of lambda-phage viruses? Styloviridae, corticoviridae, microviridae, and pedoviridae? The answer is Styloviridae. Styloviridae is a family of lambda-phage virus that are non-enveloped and has a double-stranded DNA. Question 27. What does a viral DNA becomes after being associated with the bacterial chromosome? A. Plasmid. B. Plaque. C. Prophage. D. Gene. The answer is C. In lysogeny, that is the viral DNA of the temperate phage, instead of taking over the functions of the cell's genes, is incorporated into the host DNA and becomes a prophage in the bacterial chromosomes, acting as a gene. Question 28. Vaccination was invented by A. Watson, B. Jenner, C. Crick, D. Pasteur. The answer is Louis Pasteur. So the answer is Jennifer. So in 1796, Jennifer first vaccinated an 8-year-old boy with material removed from cow, and it gives protections against the smallpox virus. So it's the work of the Jennifer is the first scientist that invented vaccination. So 29. For which viral disease vaccine has been recently developed through the use of tissue culture? A. Smallpox, B. Rabies, C. Mumps, D. Measles. The answer is D. Among the virus diseases for which vaccines have been recently developed through the use of tissue culture is measles, that is rubella. Question 30. Which of the following inhibit DNA replication? A. X-rays, B. Gamma rays, C. Ultraviolet light, D. Cathode rays. The answer is C. Ultraviolet light is absorbed by many cellular material, but most significantly by the nucleic acid, where it does the most damage. So pyrimidine diamonds are formed due to which DNA replication is inhibited and mutations can result. So question 31. Which was the first disease for which a chemotherapeutic agent was used? A. Smallpox, B. Syphilis, C. AIDS, D. Malaria. The answer is B. Syphilis is the first known disease for which a chemotherapeutic agent was used. An arsenal compound known as salvarsan was synthesized by Paul Ehrlich in 1910 to treat this disease. Question 32. Bacterial cells grown in a medium exposed to high osmotic pressure changes shape from red shape to shaped. A. Elongated, B. Irregular, C. Rod shaped, and then D. Spherical. The answer is spherical. Explanation. The high osmotic pressure prevents the cells from bursting, so rod-shaped cells becomes spherical because they lack the cell structure which imparts shape. Question 33. Tyrosidins are more effective against A. Gram-negative organisms, B. Gram-positive organisms, C. Spirochete, D. Mycoplasmas. The answer is B. Tyrosidins and gramicidin are the more effective against gram-positive organisms, where polymyxins are particularly effective against gram-negative bacteria. So these are antibiotics that target different gram-positive and negative bacteria. Question 34. Question 34. Which of the following is the most accurate method for microbial assay of antibiotics? A. Chemical and biological assay, B, biological assay, C, chemical assay, and then D, physical assay. The answer is C. A chemical assay method are generally more accurate and require less time than biological method, but they are less sensitive and caution must be used less biologically inactive degradation. Products give misleading results. Question 35. Which among the following is not an ammonia oxidizing bacteria? A. Nitrospina gracilis. B. Nitrococcus oceanus. C. Nitrosomonas europaea. D. Nitrosovibrio peneus. The answer is A. Nitrospina gracilis is a nitrate oxidizing bacteria, and they are involved in the oxidation of nitrates to nitrates. Nitrites to nitrates. Question 36. Cellulose is degraded to cellobiose by the enzymes A. Cellulose dehydrogenase. B. Hexokinase. C. Beta glucosidase. D. Cellulase. The answer is B. Cellulose, that is the most abundant organic material in plant, is cellulose. And the initial enzymatic attack by the cellulose is by cellulose, which split this long chain polymer of glucose into cellobiose, which contains 2 glucose units. 37. Sulfates are reduced to hydrogen sulfide by A. Thiobacillus thiooxidans. B. Rhodospirillum. C. Disulfotomaculum species. D. Photosynthetic sulfobacteria. The answer is C. Sulfates are reduced to hydrogen sulfide by soil microorganisms like the sulfotomaculum species, like calcium sulfate gives us hydrogen sulfide and calcium hydroxides. Question 38. The microorganisms from lakes and rivers can grow at a salt condition of A. above 1%, B. below 1%, C. 2.5 to 4%, and then D. 5%. The answer is B. Microorganisms from lakes and rivers are salt sensitive and do not grow at a salt concentration of more than 1%, whereas marine organisms are halophilic, that is the like salts. Question 39. A region of estuary that are nutritionally poor, it is more likely to find which of the following organisms? A. Viruses. B. Coliforms. C. Fecal streptococci. D. Appendaged bacteria. The answer is D. In a region of estuary that are nutritionally poor, one is likely to find the Boden and O appendage bacteria in addition to pseudomonads. So the answer is D. 40. In which of the following treatment involves oxidation of organic constituent of the wastewater? A. Final treatment. B. Advanced treatment. C. Secondary treatment. And then D. Primary treatment. The answer is C. That is secondary treatment. Secondary or biological treatment is done to absorb and ultimately oxidize organic constituents of the wastewater. That is to reduce the biological oxygen demand. 41. Which among the following group of organisms are found in the ducts of cows' mammary gland? A. Micrococci. B. Microbacteria. C. Lactobacilli. And D. Coliforms. The answer is micrococci. Micrococci such as Micrococcus luteus. Micrococcus variants are found in the ducts of cows' mammary gland and dairy utensils, and they are acid producers and are weakly proteolytic. Question 42. Which of the following microorganisms is eliminated in canned food? A. Lactobacillus. B. Clostridium botulinum. C. Mycobacterium tuberculosis. D. Coxella boneti. The answer is B. The most important organisms to be eliminated in canned food is the spore-forming anaerobe Clostridium botulinum, which is capable of producing a very potent lethal toxin. 43. Bacterial cell grown on hydrocarbon waste from petroleum industry are a source of A, fat, B, vitamin C, carbohydrate, D, proteins. The answer is D, that is proteins. Question 44. Which of the following microorganisms produce dextran? A. Leuconostoc mesenteroids. B. Streptomyces olivaceus. C. Bacillus thuringiensis. D. Bacillus polymizer. The answer is E. Leuconostoc mesenteroids is the producer organism for dextran, which act as a stabilizer in food product and as a blood plasma substitute. Question 45. The principal organisms for yogurt is Lactonostoc citrovorum, B. Streptococcus lactis, C. Streptococcus thermophilus, and then D. Lactobacillus acidophilus. The answer is Streptococcus thermophilus. Streptococcus and Lactobacillus bulgarius are among the principal microorganisms responsible for producing yogurt by fermentation. Question 46. Which of the following yeast can be used to produce microbial protein? A. Eremothecium activi. B. Candida utilis. C. Saccharomyces cerevisiae. D. Candida milleri. The answer is Candida utilis. Is yeast used for producing microbial protein, which is used as an animal food supplement, that is single cell protein for paper pulp waste. Acridine orange is which type of mutagen? Chemical compound. B. Transposons. Base analog. Intercalating agent. The answer is D. Acridine orange is an intercalating agent that can intercalate between the base pairs in the central stack of the DNA helix and distort the structures and cause subsequent replication errors. Lipopolysaccharide in cell walls is characteristic of A. Algae, B. Fungi, C. Gram-negative bacteria, D. Gram-positive bacteria. The answer is C. Gram-negative bacteria. The occurrence of lipopolysaccharide in cell wall is characteristic of gram-negative bacteria, whereas gram-positive bacteria have a cell wall that contains teichoids. Fungal and algal cell walls are very different in composition from those of bacteria. Question 9. Which of the following are present in teichoic acid? A. Glycerol residues. B. Ribitol residues. C. Ribitol or glycerol residues. D. Glucose residues. The answer is C. The teichoic are water-soluble polymers containing ribitol or glycerol residues joined through phosphodiester linkages. The glycerol or ribitol is joined to a sugar residue such as glucose, galactose, or N-acetyl glucosamine. 50. Which of the following are true for cytoplasmic membrane? Site of generation of proton motive force. B. Hydrophilic barrier. C. Hydrophobic barrier. D. Hydrophobic barrier and a site of generation of proton motive force. The answer is D. The cytoplasmic membrane is a hydrophobic barrier to penetration by most water soluble molecules because of its impermeability to protons, and the cytoplasmic membrane is the site of regeneration of proton motif phosphoelectron transfer chain in eukaryotic organism. So, ladies and gentlemen, thank you very much. This is actually MCQ in microbiology. Don't forget to subscribe and join to my YouTube community. I'll see you next.